worry. I don't have fancy videos, anything, and I realize I stand between you and a reception, so I'll try to keep everyone engaged for the next 20 minutes or so here. Okay. Just to kind of recap, and I know this has already been touched on by both of the previous speakers about how we measure ABO antibodies, just a quick review here. So, since 1901, where it's just important discovery that we have red cell alloantigens, and beginning with understanding first the ABO antigen system, of which there are actually many more on red cells, we know that if we make serial dilutions of patient sera, we can then incubate those with red blood cells. Usually we use reagent red blood cells now, although not always. And then we say, well, this is the last dilution that caused agglutination, and therefore the titer, like in this example, would be 1:16. But there's many troubles with titers, and I don't know if anyone else loves this. Is my favorite. I'm not a big Trekkie, but this was my favorite episode. So you know. But anyway, so there's a lot of variability in how we measure titers, and this is one of the things that I think contributes to inequity. So you can measure titers at different temperatures. You can measure titers in different systems, on gel cards or in tubes as the endpoint. You can incubate them and not at all. You can do an immediate spin, or you can incubate in different amounts of time. Maybe your assay uses a, different red cell to serum ratio. This is quite variable too. Which diluent are you using to make those dilutions? Peripheral blood, low ionic strength serum, actually not peripheral blood, but PBS. There's many saline. There's many different diluents. Some are commercial. They come with your gel cards. Are you using commercial red blood cells? Are you using a local panel, or using your actual donor for your ABO compatible transplant? And also, are you trying to distinguish the different isotypes of antibody, IgM versus IgG? And if you're doing that, how are you doing that? And there's different ways to do all that. So all of these things contribute to a lot of variability in how we actually perform titers in the clinical lab. And so this is a slightly older report, but if you look at one from today, it looks identical. So the College of American Pathology sends out these pop quizzes to clinical labs where we perform the same test on the same sample and report it back, and it's part of remaining an accredited lab in the clinical sphere of things. And so this is an example where you see many different results provided by the same, or by different labs for the exact same sample. It's about 120 labs, I think, in this case. And you can see that there was results reported from 1 in 16 to 1 in 2048. And that's a pretty big range. And even within the same method of tube versus gel card, there's still a huge amount of variability. And I also want to point out, this is an anti-A titer. Anti-B titers are actually not graded in this largest, really the main proficiency testing serum that is used. And if we're crossing a B barrier, as we do in infants, that means that the testing that's being done, although it's reviewed by the lab, is actually not graded for accreditation purposes. And so this is from a study that I think is one of the best studies that's out there that looks at different variability for different reasons in the way we perform titers. And this particular survey, there was 24 labs in Australia and different parts of Asia that performed testing using not only the same serum, but the exact same cells were sent out to everybody. And they received results then back from this blinded survey of 1 in 8 to 1 in 1024. And even as I have in the red box here, the exact same platform using the exact same cells and the exact same serum, the results varied from 1 in 32 to 1 in 1024. But yet these are the results that we use to make clinical decisions, big clinical decisions. This is blurrier than I anticipated it would be, but and these are data that was provided from one of our collaborators, Dr. Kathy Murphy, and this is a lot to look at, but each one of these points is an individual sample from a renal transplant patient, and then tested by 3 different ways of doing agglutination. So you don't need to, you can sift through the blurriness here to just see that this, say, for this exact patient here, maybe this result using one titer method would be this by another titer method and this by another titer method. And you can see, the main point of this slide, the takeaway is you can take the exact same sample and test it 3 ways and get different titers. So in the HLA lab, and Laurie showed that nice sort of evolution slide, and I searched so long to find like a female at the end of that evolution slide, by the way. That didn't happen easily, so it's hard to find. But and so we've had, over the course of the last, well, for over 20 years, we've been actually using solid phase antibody test methods in the HLA lab. So for those of you who are unfamiliar, this is what a Luminex sort of bead platform would look like. So we have these 100 beads in this panel, though now we can go up to 500 beads. And each of the beads is differentiated from the other bead by saying, well, this is amount of like this infrared dye versus this amount of another red dye. So when we're acquiring these on a Luminex machine, which is basically a flow cytometer, the instrument can tell apart these beads by the amount of color on each bead. And we use another color to say, well, did something attach to that bead? So, so the vendors of these products will put, say, HLA-A3 on one bead and HLA-B8 on another bead, and B51. So I just threw in random HLA antigens here, and then we can get a readout so that for each one of those antigens, we can say, oh, this is how much antibody the patient has. This has really revolutionized how we were able to provide HLA antibody support for clinical programs, especially for patients with lots of antibodies. And as I said, we've been doing this now for over 2 decades. And so you get an output that looks something like this. So if your patient had an antibody to B7, you can say, oh, this is a pretty high amount of, this is the amount of fluorescence of the HLA antibody that's attached to that bead. And say, well, there's quite a bit of antibody there. And you can do a pretty informed immune risk assessment of whether or not, if you're going to cross that antibody for your patient, roughly how much antibody is there, and make those decisions. And any of you who are working in transplantation and have these discussions with your HLA lab already understand this. So our thought then, when we were thinking about ways to evolve the ABO antibody test method, is to basically just, let's not reinvent the wheel. Why can't we do the same thing, working with the antigens that are already being produced by the carbohydrate chemist colleagues that Lori mentioned, and produce a Luminex bead-based assay that's similar. And so we did this in this same kind of a way. So we would have these basically uncoupled or naked Luminex beads attached to them individual sugars that Lori showed in that beautiful table of all the 18 subtypes of sugars. And then you get a mix of beads with which you can incubate patient serum, and you need very small volumes of this, only about like 20 microliters, which is a lot less than you would need for a titer test. And then you can measure the amount of IgM or IgG antibody that's attached to each of those beads and get a readout that gives you an amount of antibody to each of those subtype glycans. So in an example here, this is an ABO individual. Their antibody profile might look like this, where you would have, say, the A subtype 2 antibody, this much IgG and this much IgM, and so on and so forth for anti-A and anti-B. And an anti-B individual would look different because, of course, they would have anti-A antibody, but the B subtype 2 antibody, which is the relevant antibody, is low. We're still learning about some of these other subtypes for B, and if there's not time today to talk about them, but if you would have asked me later, I'd be happy to bore you to death about that. And they, oh dear, these are all so much blurred and they moved on my computer. But anyway, so this is just an example when we took this product and then incubated it, these new beads, these bead sets that we developed, and then incubated them first with just adult healthy controls to say, well, what does this look like, and what kind of a range of antibody do we see? So in anti-A antibody in healthy controls of either O or B blood type, you can see that there's a wide range of anti-A antibody to all the different subtype antigens. And for IgG, ABO B individuals had a lot less IgG antibody to A as compared to the O patients, but their IgM antibody was actually really similar. And we saw the same thing again in this also blurry picture, where we could see that the O individuals made a lot more anti-B antibody to all the subtype antibodies from an IgG perspective, but their IgM levels of antibody were really similar between O and A individuals with regard to their anti-B antibody levels. So that's interesting information that's much easier to glean using a method that is specific for isotype and impossible to measure using red cells. So, oh, thank goodness, this one is blurry. So one of the problems that I sort of mentioned to you before is that we have this reproducibility problem, even using the same platform with regard to using red cell titers. So when we took this same assay, we had done many runs over the course of time. So we had a positive control that was included in all the runs. So looking over the course of 80 different runs, the 95% confidence interval was really nice and tight, showing that it's a lot easier to achieve reproducibility of antibody testing for ABO antibodies using a bead-based platform, which we already know from HLA antibody testing as well. And then we also wanted to see, well, this works great in our hands, but how would this work if we sent this out into the world? And so we sent, just before everything shut down in March of 2020, yeah, that's right. Isn't it a blur? My goodness. Anyway, we sent out samples to 9 labs internationally. So there was labs in Canada, the US, and 1 European site, where we sent them sort of a kit of the beads that we produced, the wash buffers, the SOPs, the settings for the Luminex machine. We said, okay, run all these samples and please send us your results. But we didn't tell them what we were sending them. It was blinded. And we got back these results. And you can see this is just an example of the positive control. This is the anti-B antibody. It's really nice and tight. The A antibody is a little bit of a wider spread, but we later learned that's because we were using two different flavors of Luminex machines. And so if we were able to repeat this and use the same Luminex machine, they're not as reproducible at the upper levels of the fluorescence intensity, but the B shows this really nice tight reproducibility. So these are labs that had never run this assay before. They used assays for HLA, Luminex bead-based assays all the time, but it just demonstrates that you can really reproduce the results, not just within the same lab, but within different labs. This is really important if you're thinking about pediatric studies, as was discussed earlier, where you might have small numbers of centers, small numbers of patients per each center. You need multi-center trials that you may actually be able to reproduce these in this way, and certainly from run to run within the same center. This was, we were a bit sneaky and we sent the same sample within that sample set, and this is just showing that within the same lab, the same sample had a high reproducibility as well. So what does this mean for our pediatric patients? And Simon already discussed some of the changes that are happening with the UNOS OPTN, and I'll touch on that in a minute here too. But so how are these ABO antibodies developed then during early life? And this, you know, is especially important as we want to think about changing policy and transplanting older children across ABO barriers. And so we collected samples from the cardiac cath laboratory. The reason we did that rather than using the stored samples that we have in the HLA laboratory for children who are awaiting transplant is that these infants and children were far less likely to be transfused. We started first by looking at our patients who are awaiting transplantation, but the high use of VAS, mechanical circulatory support surgeries meant that we were finding a lot of passive antibody from transfusion, and it was making it really challenging to interpret the results. So using this population of children who are still not playground healthy, but undergoing procedures in the cardiac cath lab, not for transplantation purposes, allowed us to study this with a lot more clarity. And so what you can see here in this image is that these are children, these are blood group O children, that in their, when they're under 2, they have very little, essentially negative levels of IgG antibody, but that they have some IgM antibody developing in that time period. And then as they get older, so between the ages of 2 and 10, and 10 and 18, you start to see this emergence of IgG antibodies, and their IgM antibodies also went up a little bit in that time period. But this is a big shift here of the IgG antibodies specifically. We did try to analyze these by sex. We weren't really sufficiently powered to do that, but we hope to we will continue to look at that as we have larger numbers. And then, so thinking about the blood group O versus blood group A children and the amount of anti-B antibody, we didn't have very many ABO-B infant children, so we couldn't really do this in the opposite way. But thinking about, how much anti-B antibody is developed in comparing blood group O versus blood group B children, you can see that there's a difference in these two children. And Lori touched on this too, saying, you can't just say, well, crossing A barrier is the same as crossing B, or if your recipient is O and they're receiving an A, or your recipient is a B and they're receiving an A, is it going to be the same story? And so here for the ABO-O children, you can see that the same pattern that we saw for the development of anti-A antibody, that Ig, and it's a little different for IgM, it's a little bit slower to evolve, but you can see this evolution of antibody, and over the age of 2, O infants are starting to develop anti-B antibody. Whereas in ABO-A children, the anti-B, the IgG antibodies to B are really only starting to emerge in this age group here, over the age of 10, maybe a couple infants and children here. I guess 2 isn't really an infant anymore. And then IgM, again, a little bit of a different story than the than the O infants, but still this sort of difference in meaning that you have to think about the blood group of your child, the blood group of the donor, and not, think of it in a very individualized way, and that we, using better tools can really start to tease these things apart. So why does this matter? And Simon understands these policies far better than I do, but I put this policy up here specifically because of one reason. I just wanted to touch on this particular point. So this is the policy change that occurred in just last year, 2023. And so one of the things summarized, and I put it in the red box here on this slide, is that there's a requirement to retest every 30 days, and that the titer needs to remain less than 1/16. But if you read through this entire policy, nowhere in that policy does it say what method you need to use. So if you're using a method that doesn't enhance for IgG antibody, you may not detect that antibody. And I just showed you here on this slide that IgG antibodies are quite different, and they develop over different time points than the IgM antibody. So if your transfusion medicine laboratory isn't using a method that's going to detect IgG antibodies with sensitivity, and there's nothing in the policy specifying that you need to do that. As we are studying and considering older children for ABO compatible transplant, that's something that's worth considering, and I think it's worth having a discussion with your transfusion medicine laboratories to ask about the detection of IgG antibodies and their titer methods when we're considering transplanting older children. Oh, I think I already said all this. So yeah, because of different titer methods, inherent variability between centers, this also then creates inequity between the programs. Because if one program is using a very sensitive method and one is using a less sensitive method, one program will list that child for that ABO compatible transplant and one will not. And again, if we had standardized reproducible ways to measure these antibodies, that would reduce that inequity and it would also increase the safety. So just switching gears a little bit to typing, and thinking about this again as an opportunity and also some work that we probably need to do. We currently do typings by serologic methods. So we're inferring the typing from phenotype of red cells and then extrapolating that to say what might be on our cells and tissues. So just thinking about typing specifically, this is more relevant for ABO-A2 incompatible transplant, but also the same principle for what we're discussing here. So Lori mentioned that there's the different subgroups of A, and the most common one is ABO-A2, where about like 15 to 20% of Caucasians have ABO-A2 subgroup if your blood group is A. But the way we define that currently is by lectins. So if your red cells agglutinate with this lectin, which is essentially a piece of plant, then that means you're most likely an A1. And if you don't agglutinate that, you're a non-A1. But these are phenotypes. A2 is a phenotype, not an antigen. And so defining all these things is tricky. And ABO A1 versus A2 individuals have different typings. This is something that's about to become more relevant in Canada, as sometime next year we're going to implement ABO-A2 incompatible transplant programs as part of our kidney impaired exchange program. So this is for kidney patients, pediatric included. So stay tuned. I think there's a lot more information that will be coming our way about that. And then also thinking about, as Rory mentioned, the zygosity. So we're doing a phenotyping. We don't know if that individual is an A1O, an A1A1, an A2O, an A2A2, etc. And this may matter too, because if we're thinking about crossing ABO barriers, and there are some studies looking at the hemolysis that follows sometimes after IVIG or IVIG-related hemolysis, that if you're homozygous, that you actually have more A sugars and you have more hemolysis. So I think that's something else that we need to think about. And we've had this, again, this evolution of typing in the HLA, histocompatibility world, where we used to use serology and anti-sera to perform typing, and now we do all of this by molecular methods. But it's trickier for sugars. So when we think about sugars, we're actually typing for if we're genotyping, we're typing for the transferase, the enzyme that lives on this chromosome. But then there's also, because sugars are built on sugars, built on sugars. So then we might also need to think, well, what about the H typing, the glucosyl-transferase? What about secretor status? What about Lewis? All of these things may be important. They live on different chromosomes and different genes. So certainly the A and B glycosyl transferases, there are commercial assays available to genotype those, but it might be more complicated than that. And it's certainly more complicated than that when we think about what ends up on our cells and tissues. So when we're thinking about typing, and transfusion accreditation standards still require that we do phenotyping, and I think that's a good thing because we actually don't know the result of every one of these glycosyl transferases and what it exactly translates to for our cells and tissues, because there are actually hundreds of different alleles for ABO, which I'll show you in a moment. So when we think about this, if we wanted to really understand it, we need to know the genotype. Then we need to do flow and immunohistochemistry studies to understand, well, what does that particular allele do? And where is it ending up on individual tissues? What does the kidney look like? What does the heart look like? What does the liver look like? And then we would actually be able to use, if we had, again, better tools, we could use the ABO antibody profile to kind of as another sort of piece of that tripod to say, well, if you don't have the antibody to it, then maybe you have it somewhere. And if you do have an antibody to it, probably your immune system isn't seeing it. So these three pieces of the puzzle can really help us tease apart what do the different ABO alleles do, what are the glycosyl transferases doing, and what does that mean for our different tissues that we might want to try. So this is an example. I just took a little screenshot of the ISBT is a group that has working groups for all the different red cell alloantigen systems, including ABO. And so this table exists if you go to their website here. And this was last updated in 2017. And it's a PDF table where you can see where the differences exist. So in contrast, and I know, don't worry, I'm not going to make you really understand this. I'm just trying to make a point. So in HLA land, we have this called the IMGT database, and you can look up. So I just put an example here of the HLA-A1, O1, O1, O1 allele, and we know all these things about it. We can say like, oh, here's the nomenclature, here's this part of this group, here's a bunch of cell lines where it's been tested. You can click on every one of those, and it gives you more information. It gives you examples of like where it falls into our tables. And then also, oops, yikes. And then it goes on to tell you like, the cells that were used to, first describe this. You can click on them. Gives you a bunch of references of publications. It gives you the serological equivalent. It shows you the exon map of that particular allele. And then it gives you the genomic sequence. And if this was live, I could click on the proteins. It would show you that. So you have all this information. This is what we know about thousands and thousands of HLA alleles. So again, in contrast to a PDF table that hasn't been updated since 2017 for ABO alleles information. And these are updated, by the way, every couple months. So there's a large discrepancy between the genotyping knowledge of ABO and HLA. And so, so I'm going to circle back to that in a minute as an opportunity. The other thing I just really quickly put up here is that also another way that we can look at equity with regard to ABO blood group is something that will also be coming soon. I don't think we have a specific date yet, but we're going to be adjusting the way we calculate PRA. So PRA is the HLA measure of antibody reactivity from 0 to 100%. That also takes into account the ABO blood group. And if you haven't already seen this paper, it's a great paper, and it describes how we can then adjust a little bit for blood group when we're calculating CPRA for patients. And that will make it a little bit more equitable as well. So thinking about the opportunities, ABO-compatible transplant has obviously been reducing barriers for patients, for particular pediatric heart transplant patients for over 2 decades, but also in renal transplantation for even a little bit longer than that. And the advances in ABO antibody technologies could help us reduce these barriers if they're specific to the glycans in the heart in the case of pediatric heart transplant, or the kidney in the case of kidney transplantation, particularly ABO-A2 incompatible kidney transplant. And they can be, if they're reproducible, so it's more equitable from center to center. And that when we have new knowledge of these ABO subgroups, it may also increase donor options, because if we can define that one of these alleles that's actually present in our donor has a much lower expression of A or B glycan, then we can say, oh, well, maybe this is a better option for my higher titer patient. So what do we need to do? And this is like my dreams, so it may not currently reality. And then Lori's going to come up here too, and we're going to have a little mini rant together about ABO histocompatibility. If we can define the ABO genotypes and how they translate to organs, because this is histocompatibility, the red cells, as Lori said, they're not going to put an A red cell unit to the B person. They're just never, ever going to do that. But we do it in transplantation. We do it to increase options for our patients. And that we need an international approach to do this, so that database that I showed you of HLA, I firmly believe we need one of those for ABO, and that maybe it should fall under the ABO or the HLA histocompatibility community to do that. And that, maybe shifting again this antibody technology into the 21st century. We have done this already. We don't need to reinvent wheels. We know the process. I'm HLA old. So when I started in the HLA lab in 1991, we didn't do any of the methods we do today. And conversely, we don't do any of the methods, that we did that, I don't want to say how many years ago. We don't run those now. Like there's been this massive evolution. We don't have to change that whole pattern. We can actually take a lot of the knowledge that we've already learned and put it into place for ABO histocompatibility. And then we can also, and this was discussed a little bit earlier this morning on a few other talks, and Dr. Heller and I have talked about this, is that, the pathology phenotypes of ABO-compatible transplantation still need some of that evolution as well. We can use classic pathology and think about our BAMP and ISHLT criteria and how they may or may not be currently incorporating guidelines for ABO-compatible transplant. And what is the molecular phenotype of an ABO-compatible transplant? I don't think we have any idea. If someone in this room knows, please tell me, because I don't think we know. And I think we should try to define those two so that we can differentiate them from protein responses, as Simon already so beautifully described, that they're not the same. And so maybe we shouldn't be thinking about the same way when we're thinking about molecular ways of assessing our biopsies. So do you want to come up and finish here together with our same rant? Do you want me to present this?
Yeah, go ahead.
Okay. So I've kind of touched on this already, but new tools will help us to really drive not just the what we do clinically, but to actually help us better research ABO and other glycan responses. Perhaps this is important for vaccines work too, as Simon was describing, and that if we can reset this framework to think about ABO histocompatibility along the same lines that we think about HLA histocompatibility, then we can provide an actual immune risk assessment, which we currently don't do that. The transfusion medicine laboratory will give you a titer, but they don't say, oh, here's your risk assessment, in the same way that, like, if Esme or I are presenting an immune risk assessment to any of our clinicians, we'll say, this is the antibody, this is what we know from their previous exposure, these are the other things we know, and put it together as a picture. And that would be, I think, what ideally would be helpful for ABO incompatible transplantation as well. And then ultimately seek new clinical opportunities for safe ABO compatible transplant that expands options for patients in a more equitable way, and including HLA sensitized patients who need the most options. Did I say all that right? Okay.
Yeah, I think so. So I know we're going a little over, but
This is it. It's the last slide.
a rant. So permit me a little rant, because we're working toward this. There is a huge inconsistency in the way we deal in transplantation with ABO issues and HLA issues. And it's not to the benefit of our patients to have that continue. So for example, for technology, HLA issues are, generally speaking, use highly sophisticated technology, it's rapidly evolving, and it's globally standardized. In contrast, for ABO issues, the current technology, if you like, is maybe adequate for transfusion, but it's lacking target specificity for transplantation, as well as standardization and reproducibility. As far as clinical decision-making, HLA issues are driven by an ever-expanding scientific understanding of HLA, whereas for ABO, clinical decision-making is based on assumptions rather than any evolving scientific knowledge. The impact on organ allocation for HLA is not regulated at all. Nobody's going to come to your transplant program and say, you can't do that transplant in that highly sensitized patient. Whereas for ABO, it's highly regulated. Organ allocation is highly regulated according to ABO blood group disparity, not on any actual measurement. And the impact on risk consideration is, I think, really important in the philosophy of what we do. We all know that transplantation involves risk, and we assess the risk, and we work together with our teams to assess the risk and come to some decision making. In contrast, for ABO issues, risk is, generally speaking, just avoided. And maybe that's extrapolated from transfusion risk. And finally, the characteristics of the field. The HLA global community works together, shares practices, shares samples, tests each other. And so it's a highly evolving, amazingly collaborative world. Whereas for ABO at the moment, we're currently not meeting the needs of transplantation. And it's my view, and I think we share this view, that ABO histocompatibility really needs to live in the HLA lab. And so we'll just leave it there. Thank you. And I'm sorry we went a little bit over it.